Hello students, welcome back. Today, this is the last video of Cell, the fundamental unit of life. In this video, we will learn about the tonicity of a solution and cell division. So let's start. First, let us recapitulate the difference between plant cell and animal cell. In the last video, we read about the organelles. So from these organelles, what do we learn? What is the difference between the plant cell and animal cell? The animal cells are generally small in size. Why it is small in size? Because it is many in number. Whereas the plant cells are larger than animal cells because it is fewer in number as compared to animal cell. Cell wall is absent in case of plants, animal cell. Cell wall is absent in case of animal cell, whereas there is an additional membrane present in the plant cell, which is known as cell wall. What membrane both has? The plasma membrane. There is an additional membrane which is present only in plant cell, that is cell wall. Okay. Plastids, we learned that in animal cell the plastids are absent, but it is only present in euglena. This is the exceptional. Plastids are absent in animal cell, but it is present only in case of protozoan euglena. We know the plastids are present. Now, how many types of plastids were there? 3 types: chloroplastid, chromoplastid, leucoplastid. Chloroplastids were responsible for imparting a green color to the leaves. Chromoplastid, it imparts a bright color to the petals and fruits, to the peel of the fruits. Whereas leucoplastid, it is colorless, it stores starch. Now, here vacuoles, if the vacuoles are present, they are very small and temporary. They are very small and few in number. If vacuoles are present, generally it is absent. If it is present, they are very small and few in number. But the plant cell, in case of plant cell, it is permanent and large central sapped vacuole. And this vacuole occupies most of the space. This vacuole occupies most of its space. Okay. Now, in animal cell, they have centrosome and centriole. These centrosome and centrioles are responsible, and they only initiate and regulate cell division. They only initiate and regulate cell division. But in case of plant cell, they lack centrosome and centriole. They lack centrosome and centriole. Okay. So, this is the structure of plant cell and animal cell. Now, you can see one more difference, that is that nucleus. In case of plant cell, nucleus is at the periphery. In case of plant cell, the nucleus is at the periphery. Whereas in case of animal cells, it generally lies at the center. It generally lies at the center. Okay. This is the cell membrane. This is the nucleus which synthesize RNA. Smooth endoplasmic reticulum. Why? Because it lacks ribosome. Rough endoplasmic reticulum. Why do we call it rough endoplasmic reticulum? Because the ribosomes are present on it. Ribosome, the kitchen house of the cell. These are vacuoles. See, in this case, the vacuole is very large. And in this case, even if it is present, these are the small in number and few in number. Mitochondria, the powerhouse of the cell. It gives us energy in the form of an ATP. Golgi apparatus, flattened sac, and vesicles are there, stack off. Now, these are the responsible for synthesizing hormones and secretory hormones and proteins. Now, centrioles, which is present only in animal cell, responsible for initiation and regulation of cell division. Chloroplast, which is only present in plant cell. This is the site of photosynthesis. Cell wall, which is present in plant cell, but it is absent in animal cell. Cell wall, it is made up of cellulose, and cell wall, it is the cell membrane, it is made up of phospholipid protein. Okay. Now, we will study about diffusion. Now, we know that the molecules entry inside the cell, either through cell wall or through cell membrane. Now, when they enter, what process do they follow? So let us study. First we will study about the diffusion. Now what is diffusion? Diffusion is the movement of molecules from the region of its higher concentration to the region of its lower concentration. Now why do I have given stress on its? Because it is not like that it will flow from higher concentration to lower concentration. No, this statement is wrong. From the region of their higher concentration or its higher concentration. That means suppose you have taken two tumbler, okay, and you have somehow you have connected those two tumblers. Now what you will see is one tumbler is having a high concentration of water and the second one is having a low concentration of water. So what it will do, the from the high concentration of the tumbler the water will flow to the low concentration till it reaches equilibrium, right? Now one another example let me give you. Suppose you have sprayed a perfume on your body. Okay, you have sprayed a perfume on your body. Then what do you see? That after when anybody comes from outside, they smell, they can feel that smell that you have sprayed a perfume. Why is it so? Because that if your body is at higher concentration, the gas which is present in your body that was at higher concentration, it diffuses to the lower concentration. That means the person who has just entered in the room, so they can feel. So after few minutes or few minutes, they cannot sense it anymore, they cannot smell it anymore. Why? Because it reaches an equilibrium, it reaches an equilibrium. So I hope it is clear. What is diffusion? Diffusion is the movement of molecules from a region of their higher concentration to the region of their lower concentration, is known as diffusion. Okay, so many molecules diffuse across the cell membrane, whether it is dissolved minerals or solvent or solute, but it diffuses, right? Next we will study is osmosis. Now what is osmosis? Osmosis is the movement of water or solvent. What Osmosis is the movement of water or solvent across a semi-permeable membrane. Now this is also a process of it is also the same process like diffusion, but in this case what happened, the definition of osmosis is that the osmosis is the movement of water molecules from the region of its higher concentration to the region of its lower concentration through the semi-permeable membrane, right? Osmosis is the movement of water molecule or solvent molecule from the region of its higher concentration to the region of its lower concentration through semi-permeable membrane. Clear? Now you see these are the your water molecule and this is the solute. This is the solvent and this is the solute. This red dotted lines is semi-permeable membrane. Semi-permeable membrane means it allows only selective molecules to pass through. So you see that from this side the water molecule has passed over here. See now this from this molecule in this picture you see the water molecules concentration has increased. Why? Because it has flowed from this side to that side, from the region of its higher concentration to the region of its lower concentration. Clear? Now if you want to define the definition of osmosis related to solute, related to by taking the solute as your term, then the definition would be osmosis is the movement of solute molecules from the region of its lower concentration to the region of its higher concentration. That means when the solute concentration is lower, that means there is a high concentration of solvent or water. So we will say the movement of solute molecules from the region of its lower concentration to the region of its higher concentration. But it would be better if you all write that osmosis is the movement of water from the region of its higher concentration to the region of its lower concentration through the semi-permeable membrane. It is like diffusion, but it needs a membrane. It is like a diffusion only, but it needs membrane. Clear? Next, now what is the difference between osmosis and diffusion? Now in diffusion, flow of either solute or solvent molecules in both, it takes place in both directions. But osmosis involves the flow of solvent molecules or solute molecules. It only involves the flow of, generally it involves the flow of solvent molecules, and it is only in one direction. It is a unidirectional, but diffusion is a bidirectional. Clear? Now in diffusion you don't need any membrane. In diffusion you don't need any membrane, but osmosis requires semi-permeable membrane. Osmosis requires semi-permeable or partially permeable membrane, which allows only selective molecules to pass through. Now, number 3, it is a fast process. Diffusion is a fast process, but osmosis is a slow process. Now, diffusion can occur in both liquid as well as in gases, but osmosis occurs only in liquids. Clear? Now, when I just, as I have given you example, that when we spray a perfume, the other person can also smell. Why? Because the movements are over a large distance, because it is gas. But in this case, the movement is over a short distance. Clear? Because it is a case of fluid. Clear? Next. Now, how can substance move in and out of a cell? We have read that the movements, there is a movement of solute or solvent, but how does it take place in and out of a cell? So let's study. Now, a gaseous exchange between the cell and its external environment. Now, there is a movement of oxygen, carbon dioxide to and from the cell is carried out by means of diffusion. That means when we exchange the gases, that is in the case of respiration, the exchange of gases takes place. We inhale air which is rich in oxygen, and we exhale air which is rich in carbon dioxide. So this process this movement of oxygen and carbon dioxide to and from the cell is carried out by the process of diffusion. Clear? So gaseous substances have a tendency to move to areas where the concentration is less. We have read this in the diffusion, that it moves from the region of its higher concentration to the region of its lower concentration. Diffusion can take place in liquids and gases, both solids also. Okay, liquids and gases generally. Now, movements of water between the cell and its external environment. Now, when we are talking about the gaseous exchange, it follows the process of diffusion. But when we are talking about the movement of water molecules, it follows the process of osmosis. It is carried out by the process of osmosis. As we know that osmosis is what? It is a process in which the water moves from the region of its higher concentration to the region of its lower concentration through a semi-permeable membrane. And as all the animal cell or the plant cell, they all have a plasma membrane. Which acts as a semi-permeable membrane. So we can say that osmosis is just a special case of the process of diffusion. Why are we saying that it is a special case of the process of diffusion? Because it involves semi-permeable membrane. The movement takes place through the semi-permeable membrane. Now, let us study about the solution. Okay. Now, what we see is that if the concentration of water outside the cell is higher, okay, that means you can see in the picture when the concentration of the cell in outside is higher and the solute concentration is less inside the cell. Clear? The concentration of water is outside the cell is higher, but inside the cell the concentration of solute is less. So what will happen? The water will move from outer region inside the cell. The water will move from outer region inside the cell. That means this process, this movement of water molecules inside the cell, this process is known as endoosmosis. So which solution you can call it a hypertonic solution? When the concentration of water is more than the solute. Which solution is called as a hypertonic solution? When the concentration of water is more than solute. Now let us take an example. Imagine you have taken a dry resin. Okay, resin, kismis. You have taken that and you have kept it in a plain water. So let us take that resin as a cell. which has a solute concentration more, but solvent concentration less, and the water has more solvent concentration, right? Now after few hours, what we see that the resin got swelled. The resin got swelled. How? Because the water has moved from outside uh, to the inside the cell, and which is cell we have taken cell as a resin. Okay, so it, as a result, the water goes inside the cell, and this process is known as endoosmosis, and the cell swells. As a result, the cell swells. Clear? Let us read again. If the concentration of water outside the cell is higher than the concentration of water inside the cell gains water. The cell gains water by the process of osmosis, and this process is known as endoosmosis. This process is known as endoosmosis, and the solution is known as hypertonic solution. Now, water can move into the cell from the cell membrane, and in this case of hypertonic solution, more amount of water enter the cell, which results in swelling of the cell. Next is hypertonic solution, isotonic solution. In this cell, the concentration of water is equal both inside and outside the cell. Clear? In this case, the concentration of water is equal, is same both inside and outside the cell. So there is no net movement of water molecule. You see, the from here and here, it is equal. The concentration of water is equal. The concentration of water inside the cell and outside the cell is equal. So there is no net movement of molecule. So neither the cell swells nor it shrinks. So this type of solution is known as a isotonic solution. If the cells are put in an environment which has similar concentration of water present inside, this state allows for the free movement of water across the membrane without changing the concentration of solutes, right? So the size of the cell does not vary, neither it swells nor it shrinks, because there is no net movement of water. There is no net loss or gain of water. Next is hypertonic solution. If the cells are kept in an environment which has, in this case, what happens? The cell contains if the cells are outside it, the water inside the cell, the water content is more, and outside the cell the cell is kept in a hypertonic solution. Hypertonic solution means the solution which has more concentration of solute. Clear? The solution which has more concentration of solute. The solution which has more concentration of solute. That means outside the cell, the solute concentration is more, and inside the cell, the water content is more. Inside the cell, the water content is more, and outside the cell, the solute concentration is more. As a result, the water comes out from the cell. And the cell shrinks. This process is known as exosmosis. Why exosmosis? Because there is an exit of water molecules from the cell. We call it endoosmosis because there was an entry of water molecules in the cell. But in this case, exosmosis, there is an exit of water molecules from the cell. So this process is known as exosmosis. Clear? And in this case, the cell becomes shrink. Clear? Now, if the cells are kept in an environment which has lower concentration of water than what is present inside the cell, then due to the process of osmosis, water moves out of the cell. Water comes out of the cell, and this process is known as exosmosis and results in the decrease of the size of the cell. Now, let me give you one example. When our mother make a salad for us, okay, have you ever noticed that when we sprinkle salt in a cucumber, what do we see? That it gets shrinked, and the water comes out from the cell. Why? because that when we are adding, when we are sprinkling the salt, when we are mixing it with salt, that salt has a high concentration of solute. So it is a hypertonic solution, and inside the cell that water content is more. So we all know that cucumber has lot of water. As a result, the water comes out from the cell. As a result, the water comes out from the cucumber, which leads to exosmosis, and the cucumber slices get shrink, it gets shriveled. So this is the case of, this is an example of hypertonic solution. Okay, you have also noticed that when our parents cook fish for us, that time they mix it with salt and turmeric. Why they mix it with salt and turmeric? Because the uh, turmeric acts as an antiseptic property, definitely, and the salt being a hypertonic solution, the whatever the germs are present in the fish, it will be killed due to the, because the water will come out from the cell, water will come out from the bacteria. As a result, the microorganism will be killed. Clear? So this is why we add, we mix it with salt and pickle. Okay, now these are the, again this is the recapitulation of hypertonic, isotonic and hypotonic. Hypertonic, the what, hypertonic, the outside the water goes out, that means outside the solute concentration is more and inside the water concentration is more. Isotonic, there is no net movement of water molecules, and in hypotonic you see the water outside the cell is more, so it is going inside the cell. Clear? See, in this case, the water gets shrinked. See, the cell content got shrinked. But in this case, there is no net movement. But in this case, the cell contents accumulates its all space. So, the state of the condition is turgid. Which state you call turgid? When the cell content exerts a pressure on the cell wall. When the cell content exerts the pressure on the cell wall, this pressure is known as turgor pressure, and the condition is known as turgid when the cell is in a tight state. When it is in a tight state. Clear? Now, when do you call it flaccid or plasmolyzed? When the cell got shrink. When you place it in a hypertonic solution, the cell gets shrink. Next, what is endocytosis? Endocytosis is a process by which the plasma membrane engulfs food. See, this plasma membrane is engulfing the food and the other material inside the cell. This process is known as endocytosis. See, the plasma membrane, these are the food or the other materials which are engulfed by the plasma membrane. See, it is totally engulfed. They are starting, it is made of space and it has engulfed. This process is known as endocytosis. Next is, what is plasmolysis? Now, when you keep the cell in a hypertonic solution, the cell content gets shrinked. The cell content gets, when you keep the cell in a hypertonic solution. What is hypertonic solution? In which the solute concentration is more. I have given you an example of cucumber. In that case, in a hypertonic solution, that was when we placed it in a, mixed it with a salt, that is hypertonic solution. What do we observe? That the cell, that means cucumber, it has released water. It has released water. As a result, the cell content got shrinked. This state is known as plasmolysis. This is known as plasmolysis. So, plasmolysis is the process in which the content of the cell are away from the cell wall and shrink or contract when a cell loses water due to osmosis, exosmosis. Okay, when it is kept in a hypertonic solution. This is very important. Now, what is deplasmolysis? I told you D means removal. Removal of plasmolysis. That means, again, when you place it in a plain water, the water will move inside, and as a result, the cell content will again result in its original state. It will be again in a tight state. So, this is case of deplasmolysis. Now, we will study about cell division. Why does cell divide? What is the necessity of cell division? When we grow, the cell divides. When we got any injury, right? When we got any injury, our cells get dead. That has to be repaired. It got injury, so it has to be repaired. So for repairing the tissues and cells, their cell division is required. Then if there is any dead cell, that has to be replaced by the new cells. The old cells has to be replaced by the new cells, and for reproduction. Clear? Now let us study what is cell division. According to the theory old cells, what is cell division? When a cell is giving rise to new cell, that is only cell division. We have read this in the cell theory that all cells arise from its pre-existing cells, right? That means any cell which give rise to its new cell, that is only called cell division. So usually the cell division is of three types: amitosis, mitosis, and meiosis. Now what is amitosis? Amitosis means binary fission, which you all have already read. What is binary fission? This is the cell division in which the unicellular organism like amoeba and blue-green algae, yeast, protozoa, what do they? They first split, they elongate their nucleus, and then later on they divide their cytoplasm. So ultimately two daughter cells are produced. This type of cell division is known as binary fission or amitosis. Next is mitosis. In this division takes place in somatic cell. Now what do you mean by somatic cell? Somatic cell means body cell, the cells which we have in a body. We have 46 chromosomes, 23 pairs, 22 pairs of chromosome undergo mitosis. 22 pairs of chromosomes undergo mitosis, and one pair undergo meiosis, that is reproductive cell. Okay. In this cell what happens? The cell divides into two and produces two equal and identical daughter cells. In this case, the two cells divide into and produces two equal and identical daughter cells, and it is a continuous process. See, this is the mother cell. Okay, this replicates, the DNA replicates, then it divides. See, there is a cleavage. There is a cleavage, then it divides. Cytoplasm divides, the nucleus divides, then ultimately it produces two equal and identical daughter cells. This process is known as mitosis, and this occurs only in somatic cell. This occurs only in somatic cell, and 22 pairs of chromosomes undergo mitotic division. Next, we will read about meiosis. It is a special type of cell division that occurs in sexually reproducing organism. I told you that we have 23 pairs of chromosome. Out of this 23 pair of chromosome, only one pair of chromosome undergo meiotic division. Okay, and the meiotic division are of two stages: meiotic 1 and meiotic 2. Okay, in this case, when there is a division, it produces 4 haploid molecules, 4 haploid gametes. Okay, 4 haploid daughter cells. Okay, so meiotic 1 is equivalent to mitosis, what we read. That means this is meiosis 1. In this case, what happens? They divide into and produces two equal and identical daughter cell. See, this is meiosis 1. This has produced two equal and identical daughter cell. Now when it undergoes meiosis, there is a reductional division. Meiotic 2 is a reductional division. In this case, the number of chromosome is reduced to half. See, the number of chromosome is reduced to half. This is why we call meiotic division as a reductional division and mitosis as an equational division. Why do we call mitosis an equational division? Because it gives the same number of chromosome is passed on to the daughter cell. But in case of meiotic division, the half number of chromosome is passed into the daughter cell. So this is known as meiotic 2 or reductional division. This is the reductional division in which the daughter cell give only the daughter cell carries the half number of chromosome what the parent cell has. Clear? So I hope this is clear to you all. If you all have any problem, please kindly let me know. Till then, please watch this video, study at home, stay safe. Thank you.